Good morning, everyone. I, on behalf of Institution's Innovation Council, Tripura University, would like to welcome you all in this motivational session by successful innovators. Today, we have with us as speaker Dr. Surajit Bhattacharjee, who is Assistant Professor of Department of Molecular Biology and Bioinformatics, Tripura University. He earned his PhD from Jadavpur University on immunology and cell signaling. He is first faculty of this university to get biotechnology ignition grant and was incubated at KIT TBI and his Department of Molecular Biology and Bioinformatics. He is actively involved in research with special emphasis on innovative research. The path that he showed is continuing with other faculties, subsequently getting the big project. And the trend continues for which he was the person who started it. I welcome Dr. Surajit Bhattacharjee, sir. Over to you, sir.
Okay. Thank you, Mandakini, for this nice introduction. And as Mandakini already informed, that I am the first who did get the pay grant for the first time from Tripura University. And in this regard, I would like to give a brief idea or my brief journey, how I get the big grant and how I have proceeded to develop a new diagnostic kit. So for this, I'm sharing my screen with you. Yes. Mandakini, is it visible? Hello? Hello? Hello?
Sir, it is visible.
Mandakini? Yeah, it is visible,
Sir,
right?
it is visible. Yeah.
Okay, thank you. So I'm starting a brief introduction that how I came into this idea of developing a non-invasive diagnostic kit. And in the year 2015, uh, we had a discussion with one of my industry partner, Indian Life Science, that they are trying to develop new diagnostic kits which can be used in on spot, or which can be used as a very at a very nominal cost and through non-invasive methods. So during the discussion, we came into an idea of development of an non-invasive agglutination-based method to detect the urinary tract infection. And why I have chosen urinary tract infection, as in UTI is one of the most common infectious disease, and almost every woman in her life at one or other time face these the problem associated with the UTI infection. And in addition to this, it was also reported that a lot of boys also, or the male counterpart also getting infected through UTI during their first 10 years of life. So if we are able to develop a kit which we can use without means which we can use as a non-invasive kit, or which you can use as a non-invasive model, then it can be easily applicable to detect the UTI infection from any sample at any region. And in addition to this, the basic techniques right now which are available to detect the UTI infection are mainly time taking as well as very costly. Some of them, not all of them. Like if we want to detect the UTI, the cause or occurrence of UTI infection, then sometimes it will take more than 48 hours to carry out the culture. In addition to this, if you want to carry out, or if you want to know the to detect what are the drug through which, or what are the multi-drug resistant profile of the bacteria which are present or which are associated with the UTI infection, it also takes a long time. And there are certain non-invasive kits which we can at presently are available in the market, which we can use to detect the UTI infection, but they are not a direct method. They are through their they use an indirect strategy. Like there are kits which can use or which can detect the UTI infection through the detection of presence of nitrite. There are kits which can use to detect the UTI infection, but through the detection of leukocytes in the urine. But in my case, we have designed a kit through which we can detect the presence of bacteria in the urine sample from the suspected patient through the agarose bead surface coated antibody based agglutination method. Here, our main idea is to coat the beads, that is mainly agarose beads, with the antibody. We are immobilizing the antibody on the agarose beads, and those antibodies are mainly generated or raised against the different virulent factors or the different antigens which are present on the surface of the bacteria associated with the infection. And then if we are able to use these beads in or if we are able to use these beads and to add into the urine sample, and there we can see a clear agglutination visibility, just like the ABO blood grouping system detection method. Here it is a patient sample. We have taken the patient sample for whether our kit is working or not. So for this we have taken three different types of sample. First, it is a patient sample with bead A. Here the bead A means the beads, agarose beads are coated with Staphylococcus alpha toxin antibody, Cephorus beads, whereas the the same bead was added into the water solution, and we have taken another patient sample with bead B, where the bead B is coated with E. coli alkaline phosphatase antibody coated Cephorus beads. And here, if we observe properly, then we can see a clear visible agglutination. In the similar way, when we add the same beads with the patient sample 2, then we can found that for bead A and bead B, in both the cases we have the visible agglutination, but in when we add the both A and B, the agglutination profile still exists. So from these, we can detect the type of bacteria which are present in the urine sample, because here we are not observing any clear agglutination. But in case of the patient sample with bead B, we can observe the clear agglutination. And from these, we can detect that in case of the patient sample 1, their urine mainly contain the E. coli, whereas the patient sample 2, they contain both E. coli and Staphylococcus. Whereas in case of the patient sample 3, they also contain their urine also infected with, or their urinary tract are also infected with both these bacteria. So based on these kit, we can detect the type of bacteria present into the urine and which is responsible for UTI infection. In addition to this, we can also by changing the mode of these kit, we can also able to detect the type of bacterial type of resistance, or the antibacterial antimicrobial resistance, or the multi-drug resistance bacteria. So we are currently modifying this kit to make it more uh, usable, or to increase its efficacy, along with to use it for the other bacteria related contamination too, for detection of other bacteria related contamination. But these are under process. But so far we have detected the basic kit by which we can detect the major bacteria which are responsible for the UTI infection, that is E. coli, and as well as we are also able to detect the presence of Staphylococcus bacteria in the urine sample. So, and we have so far used these kit to detect, or used or distributed these kit to the several diagnostic kits as well as some hospitals. Like we have taken, or we have used this kit to detect the urine sample from the detect the UTI infection from the urine sample collected from the Tripura Medical College, Agartala Government Medical College, and ILS. We also use these kit to detect the urinary tract infection from the Asstt Diagnostic and Research Center in Bhubaneswar and Vivekananda Diagnostic in Bhubaneswar. And we have observed that using these kit, we can detect the we can detect the presence of UTI from 306 samples, and we have also found that almost 44 samples were negative to UTI infection. And in the similar way, our success rate is good because among the 50 501 and 515 patients, we can observe that 464 patients were UTI positive and 51 patients were UTI negative. And further, based on these observations and outcomes, we have submitted a patent in the month of February 2018, and it is still under process. So, uh, so my the so far the during the development of kit, I have faced several problems that I would like to share, so that the peoples who are into innovation, or the peoples who want to be an innovator, they can consider these points during their journey through innovation. That is, the first thing, we need to identify the right problem and the novelty of the idea. It is the one of the important factor which determines the success of our innovation. Then, along with that, we also need to identify the commercial value of idea, because if we identify a problem and if we found that this problem is novel, in spite of the novelty and in spite of identification of a right problem, if those problems is not if those problems that do not have any kind of commercial value, then we are not able to uh, apply our innovation for the purpose of commercialization. In addition to this, we also need to identify, or also need to form a good effective team member, because we need a proper coordination among the different members of the of our group. And if those members are not have a proper coordination, and if their the tuning is not good, then it will also very problematic, or it will create a problem during the process of innovation and during the process of development of those any kind of kit or any kind of new methodology. And along with that, we also need a support through which we can develop these kind of kit. So we also need a proper incubation system who can assist us to carry out these kind of innovation. And thank you. If any one of you have any kind of queries, you can contact. I have shared my contact information here. Thank you. Thank you for hearing. And I would like to thank the Institutional Innovation Council of Tripura University for giving me this opportunity.
Thank you, sir. It seems there is no one question about this. Sir, thank you for your valuable words.
Hmm.
Thank you.
Thank you.
Thank you, everyone, for staying with us, and hope we'll meet again in these types of activities. Thank you.
Hmm. Thank you.